This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 268. Recorded on June 23rd, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michelle Swanson.
Hello, everyone.
Good to see you again.
Yeah, nice to see you all in D.C.
In person. That was great. Also joining us from Charleston, Michael Schmidt.
Hello, everyone.
How's it going down there? Really hot?
It's really hot, and fortunately, when we were at ASM, it was not a super spreader event for us who practice safe microbiology, and we were wearing our masks. But I came home to Charleston, and we have 3 separate outbreaks going on at work. So we, I think, got rid of masks too soon.
And from St. Louis. Wait a minute. From St. Louis, is that
Yes.
right?
That's right. Washington University.
From St. Louis, Petra Levin. Welcome.
Hi, how is everybody?
It's a new town for me, so I'm not used to it yet.
It's a very old town.
On the river.
It's an old town. Yeah, I know. But for me, on TWiM, I haven't spoken it as much as Charleston or South Carolina or Ann Arbor or San Diego. Anyway, here we are with 2 papers for your enjoyment. And let's start with Michael Schmidt, who has something to tell us first.
Oh, yes. Well, first, Joe Handelsman tweeted out a happy birthday wish to Fanny Hess. And if you've ever used Agar plates before, you can thank her for introducing Agar as an alternative to gelatin to cultivating microbes. And she was retweeting from Jennifer Sang, who has a nice post on Frau Hess on the microbial menagerie on this day. So we'll post that link in the show notes. So it's
Michael, why is it good to switch from gelatin to Agar?
Well, bacteria can eat gelatin. And they dissolve it, so you go from a solid medium to a liquid medium, and you don't get isolated colonies.
Oh,
you get one giant plaque.
Defeats the purpose. It
defeats the purpose. And in Hess, who was a cordon bleu trained chef, introduced the Agar to Coke and her husband as a dessert glaze on strawberries. Or so I was told as a graduate student, because she was she was born in New York, and she was trained as a French chef to be a good wife to her husband back in the day. And so she learned how to make fancy desserts. So
we're talking about late 1800s, right?
Yes, 1860s.
Okay, wow.
But today we have a really neat paper, and so you can almost hear the news headline. Today on TWiM, garden compost, is it safe? So the title of today's snippet sort of gives it away. Citizen science surveillance of triazole-resistant Aspergillus fumigatus in United Kingdom residential garden soils. So for those of you casually acquainted with microbiology, you know that the fungus Aspergillus fumigatus plays a vital role in the environment, serving as a decomposer extraordinaire, responsible for global cycling or the repurposing of nutrients from its contribution in decaying plant material into soil, or as I sometimes call it, dirt. Please, no hate mail from our friends, the soil scientists. Aspergillus is a fungal genus consisting of a few hundred mold species, and it was first cataloged by an Italian priest and botanist in 1729, Pier Antonio Michele, the literal father of modern mycology, who is credited with the discovery of fungal spores. Now, this is 1729, long before Pasteur. Now, he viewed Aspergillus under a microscope, and it reminded him of an aspergillum. And since he was a priest, it's the holy water sprinkler. Any of you who are ever altar boys or altar servers have seen or have even seen Aspergillus spores under the scope can relate to how well he named this microbe. And in the show notes, I'll put a picture of Pope Francis sprinkling people with an aspergillum, and as well as the Aspergillus in full fruiting body. Now, this fungus grows between body temperatures of 37 degrees centigrade to as high as 50 degrees C or 122 degrees Fahrenheit for those of us who are metric dumb. Its canidia can survive up to 70 degrees, which is a whopping 158 degrees Fahrenheit. And you often regularly encounter this fungus in self-heating compost heaps. These are compost piles that get really warm. Many of you who have compost piles in the wintertime can often see that they can melt snow or they're giving off steam. And it's simply because the compost self-heats as the fungi are degrading the plant material and other stuff. Now, the Aspergillus spores are ubiquitous in nature, and they're very small, between 2 and 3 microns in size. And as we know from the many discussions of aerosols and the transmissions of SARS-CoV-2 over on TWiM, that size particle is in the sweet spot of being capable of remaining aloft in an air column for long periods of time. Now, everybody inhales an estimated several hundred Aspergillus fumigatus spores each day. Typically, the spore is quickly scavenged by the immune system of healthy individuals. However, in immune-compromised folks, such as organ transplant recipients, people with HIV/AIDS, leukemia, the fungus is even more likely to become pathogenic, overrunning our host's weakened defenses and causing a wide range of diseases generally termed aspergillosis. And you can also appreciate that anything that we inhale can trigger an inflammatory response. My nose has been running like a drain since DC because of all the trees that were in active pollination. And so my immune system has been sorting that out for the last 2 weeks. And severe asthma fungal sensitization is a real thing, and as well as allergic bronchopulmonary aspergillosis. Now, the significance of the small spore size of 2 to 3 microns also helps the fungus bypass our mucociliary clearance in our lungs, whereupon the spores must then evade clearance by the host immune, adaptive, and innate responses. So if they survive the host defense system, they can establish themselves in lung cavities where, left untreated, they may go on to cause chronic pulmonary aspergillosis, which the authors point out in this paper that was published in AEM. And I gave you the title, but I didn't give you the authors. And
the journal is Applied in Environmental Microbiology for those.
Yes, ma'am. And the authors are Shelton, Collins, Uzel, Al-Hamadi, Dyer, Singer, and Fisher. And they're respectively from the MRC's Center for Global Infectious Disease Analysis, the Department of Infectious Disease Epidemiology at the Imperial College of London, as well as the United Kingdom's Center for Ecology and Hydrology at Wallingford, United Kingdom, as well as the University of Birmingham and the University of Nottingham. So this is a really neat paper because they point out that in the United Kingdom in 2011, there were close to 200,000 individuals living with allergic bronchopulmonary aspergillosis. And then a smaller fraction, about 3,600, had the chronic pulmonary aspergillosis. And then the invasive aspergillosis was affecting about 3,000 patients in critical care. If that wasn't bad enough of the number of patients affected by this ubiquitous fungus, the number of patients in the UK presenting with infections that are resistant to one or more of the antifungal azole derivatives that are used to treat these fungal infections when you're undergoing care is increasing. And recall that the azole class of antifungals inhibit the fungal cytochrome P450 dependent enzyme lanosterol 14-alpha demethylase. And when that enzyme is blocked or inhibited, it stops the conversion of lanosterol to ergosterol, which then disrupts fungal membrane synthesis. No membrane, no fungus. And so these were a really important class of drugs. There's a family of azole derivatives that are used in medicine. They span the gamut of itraconazole, voriconazole, posaconazole, and these are the frontline trizol drugs for treating Aspergillus. And what we have witnessed over the last 20 years is just like in treating bacterial infections, we have watched the resistance rate go from 3 to 7% between 99 and 2001 to 14 to 20 between 2007 and 2009. And this is really pretty sad that this is happening. And so with that as prelude, let's get to the study at hand. We know that the folks in the United Kingdom love to garden, from the Prince of Wales all the way down to the person with simply a window box full of flowers. On my trips to the UK, I always marvel at how much they love their flowers and gardening. And compost plays a vital role in all of their successful gardening efforts. And the United Kingdom's government is also committed to reducing CO2 emissions by diverting waste from landfills and incineration sites to converting it to compost. So they separate their trash, and they really practice the degradation of the vegetable material into real fertilizer. And so just like getting folks to attend meetings, if you feed them, they will come. Well, when you make a lot of compost, so do the Aspergillus. Now, the aims of this study were really quite straightforward. There were 3. Determine the number of trizol susceptible and resistant Aspergillus fumigatus spores in soil samples collected from residential gardens in the United Kingdom. They're principally looking at is the average person who's tilling in their own backyard garden going to be placed at a higher risk of acquiring one of these Aspergillus infections. The second question they addressed is they characterized this cytochrome 51A polymorphisms, which is responsible for the resistance development against the azole. And then finally, they were going to ask a very straightforward question of the Citizen Science Corps. Were there any environmental variables that they could associate from the survey that was reported when the citizen scientists collected their samples, associating with the presence or the numbers of Aspergillus fumigatus spores in the 3 soil samples? So 3 questions, with each answer holding significance in terms of trying to understand this disease process that we're seeing on the increase in the UK. So how do you go about doing this? Enter the Citizen Science Corps of the United Kingdom, which is a network of trained individuals. In this case, there were 249 folks who collected 509 soil samples, and then they filled in a survey which with the aim to ascertain whether gardening activities may lead to an exposure to trizol-resistant genotypes of Aspergillus fumigatus, and if they were resistant to the azole class of antifungals. One of the things that I forgot to mention in the beginning is azoles are routinely used in agriculture to prevent fungi from attacking plants. So one of the azole derivatives that we don't use in medicine is actually used in agriculture to prevent fungi from rotting plants. So what this Citizens Science Corps found is shown on their first slide. And it's really simply a map of the United Kingdom. The geographic locations of where they collected the soil is literally plotted in colored dots. The blue dots on their graph show samples that failed to grow Aspergillus. We don't see too many blue dots. The green dots indicate samples that grew Aspergillus fumigatus, and the red dots really give us the scary danger Will Robinson sound that those soil samples grew the tebuconazole, which is the azole that is used agriculturally to treat plants against pathogenic fungi. And there are quite a few red dots. Point of fact, the Science Corps isolated Aspergillus over 5,000 times. Breaking down the data a bit further, from the soil samples, 64% of the 509 or 327 soil samples grew Aspergillus fumigatus isolates. And of those, 101 or 20% of the total samples that grew Aspergillus fumigatus were positive for being resistant to the ag azole at a pretty high concentration of 6 milligrams per liter.
Yikes.
Yeah. I mean, so again, this is a theme that we've talked a little bit about TWiM of using antibiotics in raising livestock and the danger of generating antibiotic resistant bacteria in pigs and chickens and cows. And so now even using azoles to prevent plant fungi from attacking our plants, it's pretty spooky because we don't have too many antifungals to go after. And it's really pretty spooky. Now,
and if I could just interject, that is under the umbrella of what we call One Health.
Yes.
Appreciating that what we do in the soil, what we do in our farm animals also affects human health. Yeah. How we change the environment affects climate, which can then affect the microbes, which can affect human health. We're all related.
This is a big thing. And the only environmental variable measured in this study that was found to have a significant effect on whether a sample grew the Aspergillus fumigatus or on the numbers of fumigatus recovered was the garden location from which the sample was collected. Now, the greatest concentration of Aspergillus fumigatus was cultured from a bag of manure. Makes perfect sense. You know, it's where the most organic material is. And they literally found 600 colony form units per gram. Now, remember, that's just one fungus, but the fungus makes lots of spores. And the commercial bag of manure was the highest, and it was followed by homemade compost heaps at 505 colony form units per gram. And then the commercial compost bag at 451. And commercial compost doesn't have as much concentrated nitrogen as the commercial compost. Manure has a little bit more available nitrogen simply because of how it's concentrated. And then the pot planters containing commercial compost had 321 colony forming units per gram. So the short answer to that is if you dig in your soil and you're using compost, you're really at risk to exposing yourself to a slug of Aspergillus spores. The soil samples that did not contain compost grew fewer Aspergillus fumigatus isolates, down at about 250 colony forming units per mil. And most of the samples that produced the fungal isolates were found to have been recently topped up with manure or compost, which caused me to exclaim as I was reading it, crap. I mean, I'm just going like, okay, if you are, someone who has a dodgy immune system, you really have to be careful, especially since you could send your healthy 17-year-old to dig in the dirt. And yet you can go out into the garden to look at the flowers and you can still be at risk unless it's a good windy day and you're hoping it's all blown away.
Michael, is there any epidemiological connection between any of these cases and having compost?
No, they're just looking at where it is, and they were really looking for the bulk of the paper concerns where the mutations are in the cytochrome, and I'm going to let the audience go into that detail. And there was no association for garden location sampled.
But with this data set, now that begs the question, right?
Oh, yeah.
So it can motivate those more detailed epidemiological studies.
At the least, if you have a patient who has this infection, you should ask them, do you have garden? Do you have compost? Just to try and gather that data, right?
Yeah, and maybe bring us a sample.
And remember, if you have a chest x-ray and they see a spot on your lung, it doesn't necessarily mean it's lung cancer. It could be one of these fungal blooms growing in your lung. Yeah. And so bottom lining this and summing up, 64% of the soil samples collected from residential gardens in the UK grew fumigatus. And of those, 20% of the samples grew the tebuconazole resistant Aspergillus fumigatus. So we really need to think about One Health. We really need to rethink how we're, extending the perfect garden. And
and the authors point out, like, we could use our masks that have grown accustomed to and that we have laying around the house when we're opening that bag of compost and doing that intense work. But Petra, you were going to add something.
Well, I would be interested to know, like, if the UK has a very high rate of using compost versus, say, the US, what the rate of resistance to this class of antifungals is, if there's a difference. I mean, it could be that that class of antifungals is also more common in the UK.
Well, this would be a great citizen science project that, as we transition to Michelle's STEM paper that she's going to address, this would be a great citizen science project for many of our undergraduates who are looking for exciting things to do, to ask the question, go to your gardens, collect a sample of soil, ask the question, how many Aspergillus fumigatus are there, and whether or not they're resistant to these antifungals. So, and also with big data, we can begin to look at the incidence and the GISing. We can begin to look at the Vincent's question of the epidemiological occurrences of Aspergillus in the UK versus Aspergillus in the United States. But the US has fungal outbreaks of histoplasmosis in the Ohio River Valley, cocci out west, and so we have our own issues with fungi.
And it's underfunded as an area of research. And as Michael said, we don't have great antifungals. So a wake-up call.
We have had composts for years here. We have multiple compost heaps. We take all of our vegetable material and put it in there. And I'm always mixing it up because that's the key thing you have to do.
Yeah.
Is turn it over. And I didn't ever think of this, but I've been inhaling these for years, so I must have a good immune system.
Use your masks.
Probably use your mask.
Precaution as we get into that high risk
it's the
demographic.
It's the inverse square law. As you get older, your risk increases.
I might use it anyways. There's a lot of stuff in the soil, and when you're opening the compost and just spreading it around. I mean, dogs get different things from the soil. There's no reason I think we could not get some odd things.
I look at it as I'm inoculated on a regular basis. I'm boosting my immune system. Michelle, what do you have for us?
I have a paper called NN-dimethyl dithiocarbamate elicits pneumococcal hypersensitivity to copper and macrophage-mediated clearance. And this is from the University of Arizona College of Medicine, the lab of Michael Johnson. And the authors are Sanjay Meghani, Madeline Cutcliffe, Yamil Sanchez Rosera, Chensohorina Pak, Allison Watson, Miranda Ochawa, Xin-Jiang Joyce Wu, and Michael Johnson. And they are in the Department of Immunology, the Arizona Arthritis Center, the Valley Fever Center for Excellence, and the Bio5 Institute. So the focus here is copper. And copper is, we're going to learn, is both a friend and a foe. It is classified, it's an element that's classified as one of the transition metals. Its electrons are inherently unstable, so it's highly reactive. And this makes it a great cofactor in biological processes because it can change its oxidation state, for example. So we rely on these transition metals. Our enzymes rely on them. So oxygen transport, electron transport, et cetera, rely on these. Because they're so important to many of our enzyme functions, our bodies jealously sequester these precious metals and deprive pathogens of access to them as a way to defend us against infection. And we call this nutritional immunity. Not only that, but macrophages also have ATP-dependent pumps that can dump high levels of copper into the phagolysosome, which is the compartment where macrophages do the killing of the microbes that they've ingested. But, of course, knowing that metals are used as a tool by macrophages to kill microbes, some have evolved mechanisms to protect themselves. So, for example, Pneumococcus, that we'll talk about today, encodes a pump that can get rid of copper that has gotten into its cytoplasm, and that's one of its protective mechanisms. And that's actually work that the senior author, Michael Johnson, did when he was a trainee at St. Jude's Hospital. So Pneumococcus is a great model system for studying the interplay between host and pathogen and how copper can influence that one way or the other. So the other reason Pneumococcus is such a great choice of a pathogen here is that it is a really serious public health problem. So in 2019, the CDC recognized it as a serious threat. In 1 year, we typically have 150,000 people get hospitalized with pneumococcal disease. It affects especially the very young and people at the other end of the life spectrum. Older people because their immunity naturally wanes, and then young kids, infants, pediatric population, because they can't yet generate antibodies against the gooey polysaccharide capsule that protects pneumococcus from innate defense mechanisms. Fortunately, we do have vaccines, and they are made with antigen from these polysaccharide capsules. But again, pneumococcus fights back. There is tremendous variety of the pneumococcal capsule composition out in nature, more than 100 different serotypes. So we do have vaccines against the most commonly circulating serotypes of pneumococcal capsule, but nature abhors a vacuum. So if we reduce the number of those vaccine protected serotypes that are circulating in the human population, it creates space for other serotypes to move in. So the authors here actually reference a paper showing that in Ghana, the most prevalent strains now are the ones that are not covered in the widely used pneumococcal vaccines. Not only that, but pneumococcus has acquired antibiotic resistance. So there was a study showing that 30% of cases are caused by pneumococcus that's resistant to at least one of our antibiotics. So this is a huge problem globally. In 2016, it's estimated that 500,000 people older than the age of 70 died from pneumococcal infection. So copper is important in the host pathogen interplay, and pneumococcus is a major health problem. So we are fortunate that Michael Johnson and his colleagues are using this as a model system to see if we can better understand how copper impacts that host pathogen interaction and actually exploit it for new therapies. So before I go any further, does anybody else want to add any background information?
Well, zinc is also pretty toxic. It's also one of the other nutritional elements that the macrophage is effectively dumping in, but zinc isn't as prevalent in the crust of planet Earth, so you don't see too much of it. And as the transition elements go, the more openings that you have in your outer electron shell, where you can be that schizophrenic element, where you can accept and donate electrons very easily, the more bactericidal those elements can be. For example, aluminum is extremely bactericidal, but the problem is we live in an atmosphere of oxygen, so it passivates very quickly. It forms aluminum oxide, and it no longer is antimicrobial. So the fact that the macrophage has all of this oxygen in it, because it's trying to kill bacteria, the copper reacts with the oxygen, but copper doesn't passivate. Whereas elements like silver and others will become inert in the presence of oxygen and no longer be as active. So copper is really the ideal yin and yang element that evolution has adapted on our side to kill bacteria. And bacteria, as Michelle is going to reveal to us, have cleverly figured out how to work around it.
As best they can. But then the scientists step in and say, okay, how can we tilt the balance in our favor? So this group, this is the second paper that Sanjay Meghani has published as a first author, and we'll hear more about him from Petra in a bit. They published in 2021 a study where they started with a literature search and sought compounds that are known to bind copper and therefore have potential to impact this host pathogen interaction. And I just want to credit them for taking that, being disciplined. The old saying when I was a graduate student is a week at the bench can save you an hour in the library. So from their literature search, they identified 21 compounds, again, known to bind copper, and then they screened them for copper-dependent killing of a number of microbes, including Pneumococcus. And in that 2021 paper, they identified this dimethyl dithiocarbamate, which I'm going to abbreviate from here on as DMDC, and demonstrated using growth curve experiments and some experiments in a mouse model that DMDC is a copper chelator and that it could kill some microbes, Pneumococcus among them. So in this paper, they want to now look in more detail at the mechanism. So they first verified that the DMDC will retain its biocidal activity, not only in standard microbiological media, but also in the media that we standardly use to culture mammalian cells, because it's meant to mimic the host environment. So RPMI is what they used. And fortunately, they found that the DMDC retains its activity in a variety of different media. So that cleared the way for them to continue on their experiments and try to understand how this DMDC is causing copper-dependent toxicity of Pneumococcus. So they start with a simple experiment. They want to know if the DMDC has to get inside the cell in order to have its toxic effect. So they used a really simple classic technique, which is they know that pumps don't operate at low temperature. So they took their pneumococcal cells and put them in an ice bath down to 4 degrees and then added the DMDC and asked if it's still toxic. And the answer was no. So this indicated then that the DMDC by some mechanism was being actively transported into the pneumococcal cell. So to verify that, they turned to some very sophisticated spectroscopy to actually quantify copper in the bacterial cells. So in particular, they used atomic absorption spectroscopy, which is based on the principle that transmission metals will absorb light at a particular wavelength, and the atoms will move from a ground state to an excited state. And so you can quantify this light absorbance as a measure of the concentration of copper in a sample. So using that technique, they incubated their cells either in copper alone or in the presence of two different concentrations of DMDC. And what they found is in the presence of DMDC, the cells took up 7 times or 10 times more copper than they would in just copper alone. So now they want to ask, is the DMDC an ionophore, or is it triggering uptake of all of these transition metals, or is it specific to copper? So they repeat the experiment, but now they're using slightly different spectroscopy called inductively coupled plasma optical emission spectroscopy. And again, this is based on the principle that elements have a unique spectral signature. So you can use this type of spectroscopy on a complex sample and then estimate the concentrations of different elements in that sample. So by using that spectroscopy, they were able to show that DMDC only potentiates the uptake of copper in the presence of DMDC, but not manganese, not zinc, not calcium. So it looks to be copper specific. So next, they wanted to get into the mechanism of action. They had shown in their previous paper and verified here that DMDC potentiates copper toxicity. And often that's because the copper can actually displace other precious metals, transition metals that are essential cofactors for certain enzymes. And that process is called mismetallation. So copper can bump out iron, for example, and now that enzyme won't work. So they added excess manganese, another transition metal, and asked whether it could protect Pneumococcus against a low amount of copper and DMDC. And the answer was it could, but not at higher concentrations. So mismetallation is probably one of the mechanisms of action, but clearly there are others. So next, they wanted to ask whether a brief exposure to DMDC plus copper would make the exposed Pneumococcus more susceptible to killing by macrophages. So what they did was pre-treat their bacteria in the presence of copper or copper with the DMDC for just 15 minutes and then added them to macrophages that had been activated first with LPS and interferon gamma. So that's a state that's known to be especially good at killing. And then they looked 5 minutes later, 10 minutes, 15 minutes later. And what they found is in the presence of the copper and DMDC, they saw more than 4 logs of killing within just 5 minutes by these activated macrophages. So that was clear evidence that pre-treatment of the bacteria was making them more vulnerable to killing by the macrophages. So then to ask, how are the macrophages killing these dual-treated Pneumococcus? What they did was use inhibitors that are known to neutralize 2 of the macrophage weapons of destruction, which are nitric oxide and reactive oxygen species. So when they did that experiment in cultured macrophages, they found that they could get a little bit of neutralization if they knocked out nitric oxide or knocked out reactive oxygen species, but it was really fairly modest. So they set that aside and instead decided to develop an in vitro system to ask about the mechanism of lysosomal killing. So again, they could rely on the literature, which has established that lysosomes have high amounts of zinc, hydrogen peroxide, nitrogen oxide, and each of these are known to be toxic against a range of microbes. So they created an in vitro broth system that had high zinc, high hydrogen peroxide, and NO, and then added their Pneumococcus in the presence or absence of copper with or without the DMDC. And sure enough, they found that there was killing that was dependent on each of these host defense mechanisms, the high zinc, the hydrogen peroxide, and the nitric oxide. And that's all shown in figure 4. So while they don't understand the mechanism in detail, they do see that exposure of these bacteria to both the copper and the DMDC that potentiates or mediates intake of the copper into the cytoplasm of the pneumococcus, also makes them more vulnerable to killing. They also, somewhere along the line in the course of these experiments, probably noticed that their colonies that were treated looked different. Normally, when you plate pneumococcus or other bacteria that make a polysaccharide capsule, the colonies are really kind of juicy and have this really evident capsule. So they wanted to ask whether, in fact, treatment of their pneumococcus with the copper and the DMDC was altering its capsule. And to do that, they, in particular an undergraduate that we'll learn about, did Western blots to just look at the production of capsule in the absence and presence of treatment. And sure enough, they found that there was less capsule present on cells that had been treated with the copper and the DMDC. And because we know that capsule is so important in protection of bacteria, including pneumococcus, from innate immune defenses, including opsonization by complement and also by antibody, which both mediate phagocytosis by neutrophils and by macrophages, they appreciated that any treatment that reduces the amount of capsule on the pneumococcus is going to make them more vulnerable to clearance. So they think that that is one of the contributors to this mechanism, something they'll be interested to study more. They then wound up with another experiment looking in more detail at their mouse model of lung infection by pneumococcus. And previously, in 2021, they had shown that DMDC does mediate a copper-dependent killing and ready clearance of the pneumococcus from the lung of a mouse that's been infected by the respiratory route. So here they decided to focus and look in more detail about the host response. And the way they did that was to first either expose the pneumococcus to the DMDC and the copper by themselves or together or not, and then put them by the intranasal route into the mouse. And then at particular times later, they would dissect out the lung and then digest it with enzymes and break it up and create single cell suspension of all that lung tissue. And the value of that is they could now run that single cell suspension through a fluorescence activated cell sorter, where if you use particular antibodies that are known to bind and light up cells that express particular markers of different subsets of lung cells, they could profile the population of lung cells and leukocytes in particular to ask whether exposure to copper or copper plus this DMDC was affecting the immune response. And what they found is that if they had pre-treated the bacteria with the DMDC and copper, they saw a increased number of tissue macrophages and also alveolar macrophages and dendritic cells had been recruited to the lung. So they can conclude from that the DMDC is not simply loading up the pneumococcus with copper and affecting the biology of the microbe, but either directly or indirectly, it's also enhancing the recruitment of leukocytes into the lung infection, which would be a contributor in the clearance of the bacteria that they had previously reported. So they are left with a wealth of observations that they can now follow up on with the aim of including DMDC as part of a therapeutic regimen to treat people that have infections with pneumococcus. So I thought this was a really interesting paper in that it shows how you can use really hardcore, chemistry, the spectroscopy, also knowledge of the immune system. They're looking by FACS analysis at the white blood cell population in a mouse model, and then also some classic microbiological techniques to demonstrate that DMDC, this ionophore that binds copper and potentiates its intake into pneumococcus, has some hope for treatment of pneumococcal infection.
Well, Michelle, one of the questions that came to my mind when I was reading this is it would be nice to see if the DC was turning off capsule production, or were we selecting out for mutants? Because what worries me is right now we have capsular vaccines that work at controlling the amount of pneumonia in the general population. If we use too much DMDC with copper, it may then select out mutant strains of pneumococcus that fail to produce capsule, the so-called rough strains. But what I also found interesting is
but
that
I don't think those rough strains would survive. I think they'd be easily cleared by our neutrophils.
They would be. And that's the really cool experiment that I think we're waiting to see in the next paper.
Yeah, I'm pretty sure the pneumococcal literature has got capsule mutants and shows that at least in animal models, they're easily cleared.
That's the classic rough, smooth experiment, right? It's Avery McLeod-McCarty.
Right.
It
showing
is.
DNA is the source of that property.
Yeah.
Yeah, and the other thing that I found really interesting is why lose capsule? And if you think about it, what the bacterium is doing is it's giving an electron to the copper so its pumps can throw the copper 1 over the side. So it doesn't have the same bactericidal activity. Copper 2 is much more bactericidal. And so by having a reducing environment where you're giving it electrons, you go from copper 2 to copper 1. That then allows the pumps to throw the copper over the side because the cell tightly regulates the concentration of these divalent, single valent cations. And so by going to a reducing environment, you throw it over the side. And so it may be a
so
trigger.
Connect the the carbohydrate degradation to that ability to throw electrons over the side?
Well, you need electrons. And where do you get electrons? You get electrons from NAD. So NAD becomes NADH, and then the NADH has to go someplace. And it can either go to the membrane or it can go someplace else into the cell. And
so why does polysaccharide consumption increase your NAD?
Because you're burning it. You're not putting the electrons into making capsule. You're putting the electrons into getting copper out at the sacrifice of not making capsule. It's a trade-off.
Got it.
It's a trade-off. You're effectively, cutting off your nose to spite your face, because the capsule is so important to avoid the evil neutrophil. And at the same time, the evil copper is killing you on the inside, from the inside out. So first thing you do is get rid of the toxic waste inside your house. And then you worry about the evil wolf outside trying to huff and puff and blow your house down.
Actually, I have a question about pneumococcus, which actually occurs to me now, which is it's not always in the host, right? It's actually outside the host. It might be even more likely to see copper. So maybe it is used to dealing with these one at a time. And when it gets the 1-2 punch, that's the...
I think pneumococcus, I could be wrong, but it circulates in the human population. I'm pretty sure that's its primary reservoir.
But is it...
It establishes itself as a biofilm in our upper respiratory tract normally. And when we get influenza, it does a quorum sensing thing and it de-seeds itself from the biofilm in our upper respiratory tract and then actually sinks deep into our lungs.
So when it's in our upper respiratory tract, is it seeing the neutrophils at such a high frequency? No. So it's sort of, but it might be seeing things that are in the environment, perhaps compost fumes. So
compost fumes.
Okay.
Michelle, do they speculate on how DMDC is increasing the number of immune cells in the lung?
They don't. It's possible that it's weakening the pneumococcus and you get more pneumococcal lysis, then more microbial products could just trigger more recognition by innate immune cells. But they allow that is something they'll need to study. There are a number of possibilities.
I guess the control would be, without any pneumococcus, right? Just give DMDC and see what happens. That's hard to do, right?
And one of the other things that I noticed in their methods is they were using glutamine to supplement their RPMI. They should have been using Glutamax, because in high concentrations of copper, glutamine decays to glutamic acid and ammonia, and then the ammonia can actually chelate some of the copper. So they may get different results if they use Glutamax to provide the glutamine in their medium. This was a flaw in the New England Journal paper that was looking at SARS degradation by copper. And when you work with copper, you have to know what copper reacts with. And glutamine is evil with copper because it decays spontaneously into ammonia and glutamic acid. And you can get around it by using the more expensive supplement, which is Glutamax, which doesn't decay.
Good to know. If I could return to your point, Vincent, they did in their experiment looking at the lung cell populations, they did do the DMDC alone and the Pneumococcus alone, and then they looked at the combination. And so it's not, the DMDC is not changing the population on its own.
Okay, good. That's good.
Yeah, and they also did a control that I didn't describe. It's not toxic to macrophages either.
Okay.
So there's some potential. So I'd love to hear more about the first author.
Right. So we reached out to Dr. Mengani, and he was really excited. So hopefully we're doing him justice here. This is a really beautiful paper. But so he is currently he's an MSTP student. He's completed the PhD portion, and he's a third year back in the clinic. He didn't say what his clerkship currently is, but he's doing clinical stuff. He did his path to science started in New Jersey, where he had a great AP biology teacher, which is awesome. I think that's a lot of us I did not, but a lot of people have had great biology teachers in high school, and that really, they're so important. So Ms. Donato, a shout out from Dr. Mengani. In that class, he read The Hot Zone by Richard Preston, which if you've read applications for PhD programs in microbiology and pathogenesis, that's not an uncommon feature. It is gripping. And it got him hooked on studying viruses, although I'm very happy he's moved into bacteria for this beautiful work. He did his college degree at UPenn, where he worked in Dr. Sarah Cherry's lab, where he studied Rift Valley fever virus and arboviruses. He worked with Dr. Jerome Molliston, who's actually now at WashU, an MD PhD, for helping him sort of learn about the MD PhD and get into an MD PhD program. So we're just shouting out all the mentors here. And he took a gap year working at NIH in the intramural research training IRTA program, which is a really good program. And he studied hospital-acquired infections. In the Johnson lab, he's been really happy and he really enjoys working on the bacteria. But one of his favorite things, and I think this maybe comes from his experiences with Dr. Molliston, is training younger lab mentors. So they're members. So there are 3 undergrads, Chancerina Pak, Allison Watson, and Clarissa Ochoa, all were authors on this paper. And Allison and Clarissa were experts in the blotting for capsule. So this figure, I can't remember the number anyways that we discussed about the capsule amount, that capsule blot, that was done by undergrads, which I love to see. And Chesarina became proficient in helping him perform these killing and growth curves, which are really central to this study. And he would perform a replicate and she would do her own replicate. And when they matched, then he knew that she not only understood, but that they could trust, both of them could trust their results.
Had good hands.
She had good hands, but really they could both trust their results, which I think is really excellent. He has his piece of advice, which I think is good, but hard to practice, is good things come to those who wait. So for every success he's had in life, he's had many failures or setbacks that made the success even sweeter. And there are times, and I think this is good to remember, that when he's been looked over for a position or experience he applied for and really wanted, but didn't get, something even better would come up. So this is something I think we can all remind ourselves. And then in his free time, he's a big fan of soccer, and he really likes to go to the Jersey Shore with his family, which is a great thing to do, especially if you're coming from Arizona. So I really enjoyed all his comments. And again, good things come to those who wait is excellent advice.
And thanks for supporting TWiM also.
Yes, he's a big fan of TWiM, so thank you.
Yeah. And I will bet that another big influence on his love of mentoring is his PhD mentor, Michael Johnson. So he recently was awarded the
Hinton Award.
The Hinton Award by ASM for his commitment to the next generation, and in particular, reaching out to people who are underrepresented in our field and just need a mentor to kind of explain, this is how it works, here's the roadmap. Michael started last summer in response to the pandemic, knowing how important undergraduate research experiences are to get people the exposure and the mentors that they need. He started the National Summer Undergraduate Research Project by recruiting faculty from all across the country who could set up online-based research experiences for undergraduates. And it was a terrific success and now has funding from the National Science Foundation. And he, on their webpage, he points out that it was started by a team of microbiologists who recognized the unmistakable importance of summer research experiences in helping to support traditionally underrepresented students find their love and passion for modern scientific research. And Michael points out on their webpage that he himself is a proud product of the Chicago public school system. He grew up in a single parent household. Shout out to his mother. And then after being at Duke, he got into biomedical training and has been at University of Arizona. He's now an associate professor, but he continues to work on behalf of others. He founded a Science Soundbites podcast for kids. He also has set up he works on DNA Day, which is another kind of outreach program for people, and also set up the Bio5 postdoctoral fellowship program at University of Arizona. So thank you, Michael, for all you do, not only for human health and copper, but also to support and expand the next generation.
Yeah.
We're lucky to have you.
Very lucky. That takes a lot of energy. That's a lot of different balls in the air. So very impressive. Totally deserving of that award.
And I have to say, he's got a sense of humor on his webpage. He basically describes himself that he's been killing it with copper.
That is true. And
their hashtag for the National Summer Undergraduate Research Project is no lab, no problem.
Yeah, it's a good program. So that program was picked on TWiV a couple of months ago by Brianne.
Great.
And I went and I donated to the program because I think it's such a great idea that what they're doing there. Really
I had fortunate. Through this program, I had the experience of mentoring two undergraduates last summer and really enjoyed it. It always gives us more energy than it requires that we add.
Yeah, this program has been around from the NSF for a very long period of time. When I was a graduate student, I was fortunate enough to be a TA for the laboratory section that brought in underrepresented kids from Indiana. And we taught them transduction and transformation, and they got to get their hands wet and do some really cool experiments, and just watching the light go off when they see their first results is so rewarding.
But this particular program was built by Michael and his collaborators to be virtual. So it can gain, give a lot more people the opportunity to have exposure, mentors, and learn, like, what is an MSTP program? Well, it's an MD, PhD joint program, and it's often funded. So you pay less to do that, and great opportunities.
All right. Thank you. Thank you, Petra. Thank you, Michelle. Thank you, Michael. I wanted to read one email from Joe. He writes, I am a microbiologist working in the medical device industry for 20 years. I really enjoyed TWiM 263 lavender and catheters. It is standard practice for us to scour the literature and patent filings to gain a complete view of advancements and discoveries in antimicrobials. But any nuggets from the show are a much more enjoyable process. Essential oils are some of the oldest remedies, rivaling honey, copper, and silver. In the lab, oils like tea tree, oregano, lavender, etc., can demonstrate good antimicrobial activity and better bacteriostatic performance. It has been my experience, unfortunately, that such compounds have not panned out due to compatibility issues with devices, shelf life stability, and an overall degradation in performance when it is not just the pure oil or dilutions thereof. When I am presenting to customers at trade shows and conferences, I usually start out with a quick micro 101 on how to interpret performance data. One point I really try to hammer home is to be wary of microbiological data that's presented in percentages. A product that claims to be antimicrobial may have to demonstrate minimally a 4-log reduction, also presented as being 99.99% effective. Not everyone understands log 10, so I like to offer the quick example of simply taking 1 million colony-forming units, CFUs of bacteria, down to 100 or less. While a product claim of killing 80% of microbes sounds like a great result, one should understand that it is less than a 1-log reduction. This is important because if starting with 1 million CFU, you still have 200,000 remaining, and the infectious dose may only be 10,000 CFU or less. Percent biofilm mass reduction of 80% may also sound like a great result, but how many CFU bacteria came away with that 80%, and how many are still attached in the remaining 20%? I would argue that the remaining 20% is the most important portion to remove because it's likely to be the surface-adhered microbes exhibiting a persister phenotype and ready to reestablish the biofilm shortly after the external pressure is gone. Dr. William Costerton described this wonderfully when he made the world aware of the link between microbial biofilms and chronic health conditions. Having started my work with biofilm using biomass reduction by using OD after staining with crystal violet, we struggled to get resolution between technologies and which ones had viable paths forward. We've been doing total viable microbial counts instead, using sonication and surfactant-based neutralizing buffers to recover the total viable remaining organisms after treatment. This has provided a more refined picture of performance. Fair or not, I typically dismiss any antimicrobial or biofilm disruption performance data presented in percentages. Keep
Good
up the great work because I reference TWiM and TWiV repeatedly in my talks, and hopefully I'm getting a few to tune in. This is Joe in Minnesota. So it's really funny. We have this can of something in the bathroom and it says kills 99% of all viruses. And I'm like, nope.
Not good
enough.
I never know, really, though, whether that's their limit of detection. Maybe it kills. But,
yes.
I think with the bacteria, that's a really good point because, when you study like so many, 80% sounds really good, but there's so many of them anyways. And the ones that last are the ones that can do damage. And if the ones that last are enough to overwhelm your immune system,
Well, also with bacteria, you're doing CFUs. For viruses, they're not often doing plaque assays, right? They're probably doing PCR, which is not really telling you what's going on.
And remember that when you expose bacteria to strong oxidants, the population goes from about a 0.3% persister state to well over 90%. And that's the problem with the ready-to-eat produce market.
And persisters are bugs that are just more resilient.
Yes.
Yep.
All right, let me read one more because it has to do with lavender, and maybe Michael will remember.
Oh, God. My neurons have to work.
So Inez writes, I was looking for the study data which reports the success of, I guess, lavender extract oil, LEO, right, in suppressing L-monocytogenes, assuming LEO has a bacteriostatic effect. Was there a control group used in this study which would have contained only the carrier oils and the ethanol but without the lavender extract? The carrier oils and alcohol are components of all essential oils. Being the skeptic I am, I want to see a control group for the LEO where L-monocytogenes is challenged by other components, let's say the E and the O without the L. Thank you. Inez is a veterinarian working in public health in the UK.
Good point.
So, Michael.
She certainly brings up a good point, and I do not recall, but I will go and look and then write it back in the show notes so that Ray can post it.
All right. Thank you, Michael. And that is TWiM 268. You can find the show notes at microbe.tv/TWiM. You can send your comments and questions to twim@microbe.tv. And if you enjoy our work, consider supporting us. Microbe TV is a 501c3 nonprofit. Your contributions are U.S. federal tax deductible. You can go to microbe.tv/contribute. Michelle Swanson is at the University of Michigan. Thank you, Michelle.
Thank you. Good to be with you.
Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levins at Washington University in St. Louis. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.